Okay, here we go. Here's a voice recording for your Unit 2 test, Chapter 3, which is mostly about cells, but I've interspersed a little bit of stuff on macromolecules. So we'll start with the macromolecules and work our way through just like we did all the way out to mitosis. So first things first, macromolecules. Four major classes of macromolecules. We've got the carbs, the lipids, the proteins, and the nucleic acids. So things you need to know for the test, you need to know the purposes of these, but also you should be able to recite that there are four, right, and be able to reproduce them on the test. You should know what their purpose is, like carbohydrates. Main idea for carbohydrates is that they're energy, especially when you're talking about the simplest version. They're mostly energy. When you start getting up to something like a polysaccharide, which is a big, huge carbohydrate, you have energy that can be more storage-based, like glycogen, or in plants, starch, or you can have ones that are structural. And remember us talking about this, where you have things like cell walls in plants, or exoskeletons in insects, where it's a structural polysaccharide. So mostly, carbs are all about energy. Lipids are all about long-term energy. If you're talking about just the generic fat, they could be an insulation type of thing where they insulate organs or shock absorb organs. But we also have another group within lipids, not just the fat, that we call phospholipids. And you should know a pretty good amount of detail, pretty good amount of detail on phospholipids, spelling and talking at the same time and stuff, which have this shape like this. You got a little phosphate head and some fatty acid tails, and when they get arranged in the way of tail to tail, heads out, we end up getting a phospholipid bilayer, which is the main component of cell membranes. So we'll talk a little bit more of that detail as we get to the cell membrane part of the review. And then the last group of lipids, not just, so this is fat here, phospholipids, and then there's cholesterol. And cholesterol is used for antifreeze in cell membranes, but it's also used as a source of a precursor, a building molecule for hormones in your body. Things like testosterone or estrogen, progesterone, cortisols, things like that. So there's lots of different hormones that play a part with cholesterol. Proteins are probably the most diverse of all the macromolecules. They have tons and tons of functions, from a whole unit that we'll talk about on muscles, right? So we're talking movement. We did a huge lab on proteins with enzymes. You have a section that we'll be talking about later, where they actually transport things in and out of the cell so they can act as doorways. Oops, doorways, not boreways. They can function in facilitated diffusion or possibly active transport, depending on what type of doorway you're looking at. Things like antibodies, when we cover our unit on immunity, we'll talk a lot about those proteins. And then we have, when we get to digestion, we have things like insulin that acts like a hormone as well. So there's a very diverse, and this is limited. This is not a totally full list of protein activities. And then nucleic acid is the last one, which we'll go into a little bit more detail in this class, but not a ton, is DNA and RNA. Right, so we're talking about genetic information here, whether it's the direct genetic information or worker type of molecules genetic information. That's the big things in nucleic acids. All right, next piece of chapter 2 that we kind of discussed were enzymes. You should definitely know this diagram. This is huge. The grayish structure is an enzyme. I don't know why this is going into Skype. You get to see my family. Go away, family. You get to the brown section here. I don't know if it's selected. The brown or gray section, that's the enzyme. The substrate is the thing that feeds in. So we did an experiment where we tested lactase. So lactase was the enzyme, and then lactose in our experiment was the sugar. So lactose was the substrate. It fits into the active site, which is the groove where the substrate fits in, and then out come the products. In our example, it was glucose that got formed, and another sugar, which is a simple sugar, galactose. And that was all because of enzymes. So they're real specific proteins that act as catalysts. And what catalysts do is they lower the activation energy of a reaction. So if you're talking about a reaction energy diagram, and we have reactants here, and we have products down here, we're going to make an exothermic one, you actually have to go over this little hill to get to the products. Now, generally, all reactions will move towards products if thermodynamically stable, and you have to get over this little hump here, and that's the activation energy, so the energy it costs to get to the top of the hill. So enzymes lower this activation energy because they're a catalyst, so you end up getting a curve that just has a slightly smaller, or depending on the enzyme, substantially smaller activation energy. Notice the reactant energy doesn't change, the product energy doesn't change. So in this example here, all it does is help the reaction move forward. It really doesn't change the reactants, it doesn't change the products, so that's huge. And then you should understand that there's many things that affect enzymes and their rate. If we increase the amount of enzyme, you're going to increase the reaction rate. Increase the rate. If you increase the amount of substrate, you'll increase the rate. We also talked about things like pH and temperature. They have more of a curve. So if you have an enzyme, and we'll talk more about this later, an enzyme that likes the pH of 4, if you move beyond that and you start moving into 9, you can see that the active part of the enzyme really slows down. Its activity slows down when you start going to 9, or conversely, if you move towards 1, it goes down quite a bit. Same thing is going to happen when you have a temperature 1. So things in our body might like the 98.6 Fahrenheit, so its curve is going to be peaked around 98.6. You start pulling it in or out of that temperature range, and the enzymes stop functioning. So enzymes are super duper specific, and we'll talk more about enzymes as we move through the year for sure. All right, so then we moved into Chapter 3, and Chapter 3 is mostly about the cell. So it's the most basic unit of life, and all cells have certain things. So in this example, we have a cell membrane that goes around the outside here. Right? We have cytoplasm, which is the jelly fluid. It's mostly water, but there's other things that are dissolved in there. And then you have the nucleus that holds DNA. This is the most basic things that are found in cells. A lot of cells will also have ribosomes. And all the other organelles are kind of varied in amounts. Now, I said kind of because some cells, we'll learn later, do not have nucleus at all. So when we say, oh, all cells have these, remember that there are anomalies to all these sorts of things. So like a red blood cell doesn't have a nucleus, right? It doesn't have DNA. The only way to make new red blood cells is actually to dive into what are called stem cells. We'll talk about those stem cells. Adult stem cells for blood tissue are found in the bone marrow, and we'll talk more about bones and things like that when we get to the skeletal unit. Organelles. So you do have to know all the organelles and their functions. So I have a picture here. I'm just going to kind of talk through each one. So we'll start at the top. Really didn't talk about the vacuole, so you don't have to worry about it, but the vacuole is going to store stuff. Centrally, we did talk about, because it came up in mitosis, this is where the spindle fibers are going to come out of. Remember, spindle fibers are the fibers that pull chromosomes apart. Lysosome, hydrolytic enzymes, so this is going to be kind of like the garbage disposal. It's going to break down anything that it kind of comes and fuses in contact with. So there are diseases or functions where your body will purposely puncture this, releasing the hydrolytic enzymes, and the whole cell destroys itself, and that's called autophagy. Just in case you were looking at a question off your mimic. Right? Nucleus, purpose, store the DNA. Right? That's the main purpose of the nucleus, keep that DNA protected. Nucleolus is another dot on the inside there, and the purpose of nucleolus is to make ribosome components. Right? So it's all about ribosome production. Cytoplasm, water-based, right? Keep the cell hydrated. This is going to be the thing that loses or gains water for the most part when we're talking about osmosis. Mitochondria, all about production of ATP via cellular respiration. Those of you taking AP Bio, you're going to have a huge unit on that. Secretory vesicles, these are things that just kind of like what it sounds like, secreting. So these are the vesicles that might come off something like a Golgi, which we'll talk about in a second, and they will go to the cell membrane and then release their contents out of the cell, maybe into the bloodstream or into a duct, or into the duct would be going into some sort of internal compartment, some lumen, so a digestive canal of some sort or something along those lines. Peroxisomes contain H2O2. Purpose is to detoxify compounds, so something that might be bad in your body. Peroxisomes will take in that component, detoxify it so that the cells and the rest of the body does not get harmed. Golgi is kind of like UPS. It's the packager. So it gets the products from the endoplasmic reticulum, specifically the rough endoplasmic reticulum, packages them up, throws them in a secretory vesicle, and then punches it outside the cell so that it can be used. Endoplasmic reticulum, there are two types. There's smooth and there's rough. The rough is rough because it has ribosomes on it. And ribosomes are all about making proteins, so I'm sure you understand that. Smooth ER is going to be more about making carbs and lipids. So those are the organelles that are mostly on your test. There are some other ones that are found in some of the book. We won't focus on them as much, so they're not on the test. That being said, moving on to the cell membrane. So you got to know how the cell membrane's put together. Two big things in the cell membrane. This picture is including a lot of stuff that you really don't need to know for the test. Big thing is that you need to know that proteins, right here, fused into the cell membrane are going to act like doorways. I know that looks like a B. I don't know what was happening there. So those doorways are going to let things in and out. So if it's facilitated diffusion, right, it's going to possibly let things go in down the gradient. If it's active transport, maybe this acts like a pump and pushes things out or in, depending on what we're looking at. Those proteins are going to be doorways, though. And then the phospholipids, which are the little circles with the tails that you see here, and those act as the barrier, right? So keeping outside things out and inside things in, so that if we do let things move, we have control over it. The only things that really move through the cell membrane easily are gases that don't have charge. No charge. Which is a majority of the gases. So things like CO2, O2, N2, right? These are the gases that you're constantly inhaling and exhaling. Those things are easily diffused through this cell membrane. Water is another one that kind of moves freely, but if you go into a higher level, you'll learn that there are special proteins here that are called aquaporins that actually help out with water's transport. And water's transport, remember, is osmosis. So let's talk about passive versus active transport, and then we'll get into some examples of osmosis, and we'll be moving on. So passive means that we're using no energy, so we're going to go from a high concentration to a low concentration down the gradient. Okay, so no ATP required here. There are a couple examples of passive transport. There's just straight up diffusion, which you really don't need a membrane to do. It can just diffuse, so a smell that diffuses across the room. You could have facilitated diffusion, and in facilitated diffusion, it's still going down the gradient, but you need a protein in the membrane to help it out. Right, so you need that doorway to allow that molecule through. And then there's osmosis, right? And osmosis is the basis of our picture down here that we're going to talk about. Active transport is exactly the opposite. We're going to go from low to high. So that takes energy, or ATP, to do this. Some examples that we talked about are endocytosis and exocytosis. Okay, both of those are active transport. It takes the cell energy to push things out or pull things in. Okay, and another example of endocytosis is called phagocytosis, which white blood cells eat with. All right, so let's talk about this osmosis problem here. You're going to have something similar like this on the test. So you need to look at the diagram and be able to pull up very important vocab with it. So when we look at right here, sugar molecules are the blue. So we look clearly on the right-hand side, there's less. So the terms hypo, hyper, and iso all are designated about the tonicity, right, or the amount of stuff dissolved. So hypotonic is referring to less stuff dissolved. So that means that the other side is hypertonic. Now, if those molecules cannot move through the membrane, so selectively permeable membrane, some things can move through and some things cannot, then it's water that actually has to balance out the situation. So remember, water likes to go where the party is. There's more particles on the left-hand side as far as sugar is concerned. So the water will go from the right-hand side towards the left-hand side. That's what you're seeing in this image here. So if we had scenarios where, let me do a problem here. You have a blood cell that's sitting in water, and in that blood cell there's 30% sucrose and only 5% sucrose outside, and sucrose can't move. Well, then we have a problem here. We got to figure out if sucrose can't move through, then it's water that's got to balance it. So in this scenario, then we end up having water space availability. Right? This isn't truly how it works out chemistry-wise, but water space availability would be 95, right? And here it would be 70. So water is going to go from a higher concentration to a lower concentration because we're focusing on water when we're talking osmosis. Now, when we're talking terms of tonicity, remember, we're not talking about water. We're talking about the sucrose that's dissolved. So this is a low amount, so this gets hypo, even though there's lots of water. Here it's hyper, so you would think, oh, high to low, hyper to hypo. No, that's not how it works. Osmosis is the diffusion of water, so that's why I'm saying make sure you kind of write out the space available that's left over for water, because that's going to help you guide of which way the water is actually moving in these types of problems. Okay. Mitosis, goal and process. Mitosis is all about dividing the nucleus or dividing the DNA. All right, so we want to make identical cells. So if a cell has 24 chromosomes, which is not you, if you actually have 46 chromosomes, but if you have 24 chromosomes in a cell, after we do mitosis, we should make two identical cells that have 24 as well. Just like in you, 46, we want to go to 46 and 46. All right, that being said, mitosis is small wedge of a cell's life. This is a cell's life. So the actual dividing of DNA is mitosis. Everything else is what's called interphase, right? As you can see on this ring here. Interphase is everything else. So if I ask, when is DNA replicated? Interphase. When does the cell make proteins? Interphase. When does the cell excrete proteins? Interphase. When are fats used? Interphase. When does the cell divide? Mitosis, right? So just make sure you understand mitosis is simply about the division of DNA. And to jump in with this, during telophase, right, you get this process of cytokinesis, which is actually the dividing of the cytoplasm. So pulling the chromosomes apart is mitosis, but the actual division of the cytoplasm is cytokinesis. So both of them have to happen if you're going to make a cell that ends up having each cell having two nucleuses. Okay, and you have to know the phases, obviously, right? And if you want me to, if you want me to go through the phases, actually, I should do that real quick for you. Go away. Stupid board. So it's PMAT. Right, so prophase, metaphase, anaphase, and telophase. So prophase, you're going to just start seeing chromosomes showing up in the nucleus, but the nucleus might start dissolving. Metaphase, they kind of start lining up in the center of the cell. Right, anaphase, you're going to start seeing the chromatids getting pulled apart. And then telophase, you're going to start seeing two nuclei and the chromosomes starting to kind of unravel again. Right, look through those pictures that you have. I give you that worksheet that has all those pictures on it. That'll help a lot. All right, so protein synthesis real quick. Purpose of protein synthesis is obviously to make a protein. This is all going to happen majority at the ribosome, right, where the actual assembly occurs. But keep in mind that there are some things that happen in the nucleus, and then there are some things that happen at the ribosome. You need to know it goes DNA to RNA, right, and then it goes RNA to protein. And these have two different designations. So when you're talking about DNA to RNA, this is transcription. Like I said, it happens in the nucleus, whereas this is translation. And that happens at the ribosome. So this process happens first, this process happens second. You need to know that there's three different types of RNA. There's mRNA, tRNA, and rRNA. Those are not the only types of RNA that we know of in biology, but they're the only ones that we're focusing on for this section here. So the tRNAs are the ones that are bringing in the pieces of protein, which are amino acids. The rRNA is what kind of makes up this ribosome here. And then the mRNA is the actual copy, so that gets manufactured first here in the transcription process. Okay, so that is protein synthesis. Yeah, and that's it. So if you have questions about any of this stuff, send me an email this evening or come in tomorrow morning and I'll help you out with stuff. I wish you the best of luck and you'll ace this test.